Today we're going to be talking about gene expression, so we're going to focus on two central ideas, those ideas being what is the central dogma, and the second being regulation points. So the central dogma is a process where your phenotype, or what your physical features look like, is determined by your genotype. And the way that works is through this process that's listed below. It's where your DNA determines your protein, and so that happens from DNA. There's replication. You learn about a couple key enzymes that come into place, such as DNA polymerase and things like helicase. You also learn about transcription, where DNA becomes messenger RNA, and this is based off of base pair rules. And lastly, translation, where messenger RNA becomes proteins. And some of the key factors here are things like ribosomes, and we learn about things like the codons and the genetic code. And so we'll break down what happens in each step, from replication, transcription, and translation, and we'll also talk a little bit about regulation. So first off, what happens during replication? So if you recall, what does DNA look like? DNA is usually double helix. It's double-stranded, and we tend to have 4 bases, A, T, C, and G, standing for adenine, thymine, cytosine, and guanine. From there, we want to cause replication, so we do that using first something called helicase, which unwinds the DNA. Once it's unwound, we now have it similar to a ladder. We then can separate the DNA, and we'll use something called DNA polymerase 3. It's named DNA polymerase 3 because it was discovered third, but it's actually the most key replication enzyme. So an enzyme is something that can build and break, and DNA polymerase 3 is responsible for most of the polymerization. So using the base pair rules, it knows that every time it sees an A, it plugs in a T. Every time it plugs in a C, it adds a G. Some words that describe this are that it is semi-conservative, meaning it uses the old strand as a template. And from there, after DNA polymerase 3 happens, we also have another DNA polymerase 1 that double checks it. And this is kind of our double checker. This lowers and decreases mutations. And so if you're thinking about the main ideas for replication, the idea is that we unwind and we add using the base pair rules, and we double check. From there, we end up with transcription. So now we have the DNA that's been stored for mitosis and meiosis, and now we'd like to go ahead and make RNA. So this is movement from the nucleus, or the center of the cell, the brain of the cell, to the cytoplasm. And so the way that this works is, again, based off the base pair rules. And the base pair rules are slightly different now, because instead of thymine, we now have uracil, which is a RNA, or a ribonucleic acid. So A and U now, and we still have G and C. So what happens during transcription? We have a similar process where we have a helicase that unwinds the DNA. You have binding proteins that hold it open. And from there, we're able to make a single-stranded RNA. So what started out as a double-strand DNA, from there you want to end up with a single-stranded RNA. And this is specifically called messenger RNA because it carries the message. This is able to move out into the cytoplasm. Messenger RNA is single-stranded. And because it's single-stranded, it can move through nuclear pores. And moving on. The last step in this process is called translation. And translation is probably one of the most interesting parts. It's the idea of turning your genetic code, so turning your mRNA into a protein, or a polypeptide, made of many amino acids. So what happens? You have your mRNA, that's carrying the message. It's exited out the nucleus through the nuclear pore. We have these other things floating around called tRNAs that kind of look like clovers or keys. They contain something called an anticodon. So if we take a group of three is called a codon, and they could be things like AUG. AUG would be the start codon. It's one of the key ones. So opposing that would be UAC. The anticodon on the tRNA would carry a specific amino acid on top. In this case, our amino acid for this would be methionine. And so there's a statement that the genetic code is both specific but redundant. And what that means is that each codon codes for a specific amino acid, but it's redundant in that there are only about 20 amino acids. So we have some replication, we have some redundancy, some repetition of things that wouldn't happen. So we have the start codon. As we move along, this is all composed up by a ribosome. So we have mRNA, we've got ribosome with different sites, and we've got tRNA entering and exiting and dropping off a variety of amino acids. And so this process repeats and repeats, and eventually you end up with a protein. So that was kind of the down and dirty of what happens. So how do you regulate all these processes? So regulation of gene expression can happen at any point, but it mostly happens either at transcription or translation. So in terms of transcription, one form of regulation would be something called a transcription factor. You cannot cause transcription without the RNA polymerase, so we have transcription factors that recruit RNA polymerase. That recruit RNA polymerase. You also have an enzyme called dicer that will shred mRNA. And this is kind of a way to prevent expression. You also have kind of time lapses or the enzymes that will break down of mRNA. During translation, that kind of goes in there too. If you can prevent mRNA from being transcribed, you can also have factors, so limits on amino acids. You could have blocks in your ribosome. But there are different ways to stop and start gene expression. So in summary, at the end of this lecture, you should have been able to answer two major questions. What is the central dogma? Or how does our genotype affect our phenotype? Or another question would be that how do we make proteins? And the other question is how do you regulate this process? You can find more details in your book or from another lecture, but this was just kind of the down and dirty version of gene expression. If you have questions, please feel free to email me. Also, don't forget to refer to your textbook, each other, and peers. Hope you have a great day, and thanks for listening, and I will talk to you soon. Have a great one. Bye.